Small baby steps. There you go.
Yeah, so the troponin and CK were both elevated there.
Medical alert, adult level 1 trauma, here now.
5-year-old male.
Watch it.
Alright, we're gonna be in the trauma room 5. We have for action.
Yeah,
Yeah,
I was about
6
Alright,
on the count of 3. Here we go. 1, 2, 3. Okay, I can hand you one right here.
Okay, so let me know when you're
Alright, let's get some lab orders in. I want a CBC, a CMP, a lactic acid, a type in screen, an ICAL, a PT, and a PTT ordered.
Okay,
I'm going to start an IV on the right
You
knee.
gotta
Give him some anesthesia
Alright,
here.
Mr. Smith, hold still. Big poke here.
Can you hand me
Alright, we
another
get the
package?
orders.
I'm getting the orders in
Okay,
here.
we'll probably have to cut him morphine.
yeah.
Can you
Yeah, I
pass me
have
the
it
clips?
running on this one.
Okay, I'm gonna need to move
You got
this
two
for a
more
second.
orders.
Move this tube
I
in
would
here.
got
double
a
check
slight
with
state
Steve.
of
O2
shift.
saturation is
5
quite
minutes
low.
Okay,
Alright,
just let
suction.
me know.
Okay, can you give a 5
wait,
milligram
I'm
dose?
gonna-
ER, this is Kelsey.
Hey, it's Roger from chemistry. I've got a critical result on patient Brick Smith in trauma room 5.
Okay,
Hold it down.
Up.
Now, pull a little
I need
higher.
another
Lactic acid is
Hold this part
4.5.
here. A little higher.
Lactic's 4.5. Copy that.
Alright, this one's 2 inches to the left.
Pass me the clamp.
ER, this
Is
is
that it?
Kelsey.
Hi, this is Sally from Hematology. I've got a critical result on patient Bricks.
Higher. Right there.
He's bleeding out again on this side.
Hemoglobin is 6.1.
Let's get 2 units of blood going.
Often hidden behind the scenes, yet so important to the clinical care of a patient, lies the department of diagnostic specialists, on whom doctors rely on heavily for answers. These professionals are experts in the scientific field, working diligently to provide quality test results on patient specimens. This is the movie about medical laboratory science, a Brickfilm documentary.
Morning, Jeff. Morning, Bill. Hey, Paul.
Good morning, Jeff.
This medical laboratory scientist clocked in and is ready to begin his work for the day. He will be working in the hematology department, the section of the lab that deals with the analysis of cells, coagulation, and in some cases, urine. The fundamental and most basic test run in hematology is the CBC, or complete blood count. The basis of this test is a hemogram, a numerical set of values that summarize the quantity of cellular elements in the blood. A hemogram includes a white cell count, red cell count, platelet count, hemoglobin, hematocrit, and a few other parameters such as the red blood cell indices, which give the clinician information about the size and hemoglobin concentration of red blood cells. The provider may order a differential along with the CBC. A differential is the percentage breakdown of each type of white blood cell, along with a morphological analysis of the red blood cells. The first step in running a CBC is to count the cellular elements, which is often done via an automated analyzer. Watch as the technologist loads a rack of blood samples onto the analyzer. The analyzer takes in the rack, scans the barcode of each blood sample, and begins running the cell count. The principle behind these instruments, called Coulter counters, is electrical impedance. The basic Coulter counter consists of 2 chambers, filled with a conductive fluid. The chambers are separated, but contain a small hole between them, called an aperture. Each chamber has an electrode, and the instrument measures the current passing through the fluid. A small sample of blood is then added to one chamber, and the fluid is allowed to flow from one chamber to the other. As the cells pass through the aperture, they temporarily disturb the electrical current, which the instrument measures as a spike in resistance. The larger the size of the particle, the greater the peak in resistance is measured. Once all the data points are gathered, the computer then analyzes the sizes of the particles and categorizes them into white cells, red cells, and platelets based on preset thresholds. Modern instrumentation often uses flow cytometry methods to more accurately categorize the cells by measuring unique light scattering abilities of various cells. Hemoglobin is measured by breaking the red cells in a separate chamber and measuring the light absorption of the remaining red solution. The instrument may also run an automated differential. The technologist running the automated cell count must carefully review the results. The scientist's educational background and training are crucial in this step, as any discrepancies or questionable results must be explained and verified. Let's take a look at this hemogram. The MCHC, which is the parameter that shows the average hemoglobin concentration in each red blood cell, is 44.6. which is physiologically impossible. The technologist suspects that the patient's blood has cold agglutinins. Cold agglutinin disease is an autoimmune disease that causes red blood cells to cluster. When this occurs, the analyzer is fooled into thinking that the red blood cells are very large and few. This causes the calculated hematocrit to be falsely decreased, which in turn causes the MCHC to artificially inflate. The technologist decides to try an experiment by warming up the tube of blood to see if the cold agglutinins will dissipate. Sure enough, they did, and the MCHC is now back to normal. The results can now be released to the provider. If the instrument detects a discrepancy with the results, or produces a questionable value, or runs into some kind of other error, it may flag the results, prompting the technologist to manually make a blood smear and look at it under the microscope. The technologist unloads a sample, places a drop of blood on the glass slide, and smears the drop with another slide. Once the blood is dry, he then floods the slide with methanol fixative, followed by an eosine stain, followed by a methylene blue stain. And ending with a rinse. The slide is now dry and ready to be looked at by a technologist using a high-powered microscope. The technologist finds a segmented neutrophil, a type of white cell. These granulated cells are often the first responders to infection and can ingest pathogens like bacteria. This is a bound neutrophil, an earlier version of the segmented neutrophil which is not fully mature yet. The technologist now finds a lymphocyte. Lymphocytes show up as smaller white cells and contain a dense round nucleus with a sky blue cytoplasm. These are the cells that can become antibody-producing B cells, or a variety of T cell types, both of which are heavily involved in the immune response, as are other white blood cells. This is a monocyte, a white blood cell that can become a very large macrophage in body tissues. Macrophages ingest debris, infected cells and pathogens, ultimately functioning as the janitors of the blood. Here is an eosinophil, a white blood cell with large pink granules. These cells are often involved in allergic reactions and parasitic infections. This is a basophil, a rare type of the white blood cell with large purple granules. These cells are also very involved in allergic reactions and protect the body against pathogens. Their granules can release histamine, which dilates blood vessels and allows other white blood cells to more effectively enter infected sites. The technologist now discovers a blast, an alarming finding. These are very early and immature cells normally found in the bone marrow. Finding these cells in the blood often means the patient may have a bone marrow problem or a leukemia, a deadly blood cancer. As a part of the differential procedure, 100 white cells are counted and categorized. Afterwards, the technologist does a platelet estimate and ends with a red blood cell morphology report. The shape of the red blood cells and any noted inclusions can be of great diagnostic value to the clinician. For example, teardrop-shaped RBCs are often found in myelofibrosis. Sickle cells are often found in sickle cell anemia. Large oval RBCs are characteristic of vitamin B12 deficiency. RBC fragments frequently show up in disseminated intravascular coagulation. Basophilic stippling is seen in lead poisoning. RBCs with bulb-like projections are seen with liver disease. There are many other shapes and forms of RBCs that the technologist can recognize. The technologist also notes any unusual features of the white blood cells and platelets, and the presence of parasites in rare cases. Coagulation testing is an essential part of hematology. The body's mechanism to stop bleeding involves a very complex system of coagulation factors that, if triggered, results in the formation of a clot. The clot is made up of platelets and is reinforced by fibrin. Patients who are on blood thinners or anticoagulants rely heavily on coagulation testing. In hospitals, such tests are often used to monitor patients on a heparin drip. Blood drawn for coagulation testing is drawn in a blue tube. Blue tubes contain sodium citrate, which binds and inactivates calcium, an important ingredient in coagulation. Once the blood is ready to be tested, it is incubated with the proper reagents and receives calcium to activate the coagulation process. You are, in essence, putting the blood's coagulation cascade on a giant pause and resuming it in the test tube. The instrument measures the time it takes for a clot to form and reports the result in seconds. Different coagulation tests measure different parts of the coagulation cascade. Common tests include prothrombin time, partial thromboplastin time, thrombin time, and fibrinogen level. Technologists must ensure samples are properly collected for coagulation testing to be accurate. For example, filling a blue tube past the fill line causes an inaccurate result since excess calcium in the blood overwhelms the citrate additive in the tube, causing the coagulation cascade to slowly resume in the background. Samples drawn too forcefully or too long may clot quickly in the tube, making the sample useless.
Another bad spec.
The urines bench is yet another important part of the laboratory. In some labs, this bench is part of hematology, as in this case. The fundamental test done here is called urinalysis, which consists of a panel of chemistry tests done on the urine, as well as a microscopic analysis of larger particles, such as cells. The technologist loads a rack of urine samples onto the analyzer. The first segment of the analyzer performs the chemical portion of the panel using a dipstick. These tests include nitrate, bilirubin, protein, leukocyte esterase, glucose, and a few others. The samples then move on to the microscopic segment, which passes the urine through a camera, taking many pictures of the various particles that pass through. A computer then sorts the pictures based on what it predicts the particles are. It is the responsibility of the scientist to review those pictures and reclassify the particles as needed. If it is not clear what the particles are, the technologist may choose to manually analyze the urine under a microscope.
Looks like granular casts. White blood cells, red blood cells. Ooh, looks like some yeast.
Urine tests may provide doctors with clues as to what the patient may be sick with. For example, the presence of renal tubular epithelial cells in urine may indicate the patient has kidney damage. The presence of many white blood cells may mean the patient has an infection, and so forth. In hospital laboratories, the hematology department may also perform cell counts and differentials on body fluids. Occasionally, a crystal analysis may be ordered on joint fluid. A special polarizing microscope is used for this test. If a patient has gout, Sharp needle-like crystals appear yellow when parallel with the axis of slow vibration, and blue when perpendicular to the axis. Chemistry, a critical department in the laboratory responsible for the analysis of chemical compounds, ions, and biological molecules in samples from patients using sophisticated instrumentation. Chemistry analyzers can measure quantities of many different kinds of analytes found in blood. Testing often requires the serum or plasma portion of blood. Serum is the fluid portion of the blood devoid of all cells. Plasma is like serum, but contains platelets. Serum and plasma can be separated from cells using a device called the centrifuge, which spins the tubes of blood at a high RPM, forcing the heavier cells to the base of the tube, while leaving the lighter serum or plasma portion on the top. It is critical that the operator balances the tubes on the centrifuge. Otherwise, a misbalanced centrifuge can vigorously rattle.
Shabloop. Loy.
When blood vials are properly spun down, they are ready to be placed on the analyzer. Watch as this technologist loads the spun sample on the benchtop analyzer for a procalcitonin reading. The most frequently used tubes in chemistry are the mint green, dark green, red, and gold tubes. Mint green and gold tubes have a gel in the tube, which keeps the plasma or serum separate from the cells after centrifugation. Dark green and red tubes need the plasma and serum separated manually with a pipette after centrifugation. Green-colored tubes contain lithium heparin, an additive that prevents coagulation of the blood. Red and gold top tubes don't have any additives and thus require the blood to clot for 30 minutes before being spun down. Some labs may utilize an automated chemistry line. Watch as this sample travels down the track. The barcode is scanned, and the line sends the sample to the centrifuge module to be spun down. Samples are placed on the centrifuge, balanced, and spun down. Once the sample has been centrifuged, the line sends it to the main analyzer for testing. In the testing module, the probe is washed. Then the sample is aspirated, placed in a reaction well, probe gets washed again, appropriate reagent is selected, and a spectrophotometric measurement of the reaction is taken. This particular instrument has a chemistry module and an immunology module. The chemistry module utilizes spectrophotometry for much of its testing. The idea behind spectrophotometry is that substances in the sample absorb specific wavelengths of light. When the reagents react with the analyte we are trying to measure, the product of the reaction can absorb a specific wavelength of light. The higher the concentration of our analyte, the more the product is created, and the more light is absorbed. A sensor on the other end detects how much light passes through the sample and calculates the concentration based on brightness. This is the immunology module of the analyzer. Samples with tests that utilize immunological methods are sent here. Let's examine the principle behind the immunological testing in this chemistry analyzer. These are antibodies. Antibodies are biological molecules that the body's immune system creates when it believes a particle is a threat. An antibody has two arms which can bind a very specific molecule, such as a capsid protein of a virus. Scientists found ways to harvest antibodies that can bind the target analyte that we are trying to measure. For example, let's say we are trying to measure the amount of brain-type natriuretic peptide, or BNP, in a plasma sample. The antibodies we will use here specifically can capture BNP molecules and no other molecules. In theory, we can add a second antibody that is chemically linked to an enzyme. The secondary antibody will also bind the BNP. We can then add a substrate to the enzyme, which will allow the enzyme to generate a product that can fluoresce and generate light. Based on the amount of light that is produced, the analyzer can calculate the concentration of BNP in the blood. But there are problems with this concept. First of all, any free secondary antibody can react with substrate, producing extra light or signal. Additionally, there can be substances in the plasma that can interfere with the signal. To circumvent this, scientists developed a clever solution. They chemically couple the primary antibodies with magnetic beads, which are represented by these white particles. The plasma is added, and BNP molecules bind the antibodies on the magnetic beads. A magnetic field is applied to the sample, and the beads with our target molecules are pinned down tightly while the rest of the solution is drained and washed with a buffer. The secondary antibodies linked with enzymes are then added and allowed to bind the BNP particles, effectively forming a sandwich. The magnetic field is applied once again, and any unbound secondary antibody is washed away. The reaction cuvette is then flooded with substrate for the enzyme, and the enzyme converts the substrate to a fluorescent end product. The amount of light that is produced is proportional to the amount of BNP bound, and is measured by a sensor on the instrument. Other benchtop analyzers include blood gas analyzers, which can accept syringes with arterial or venous blood and measure the oxygen and CO2 saturation, pH, lactate, sodium, potassium, and other electrolytes. A decent portion of a technologist's day consists of running QC, calibrations, and performing maintenance on analyzers. Instruments are periodically calibrated to ensure correct measuring. QC, or quality control, are samples of known concentrations that are ran on the instrument to ensure proper function of the analyzer. Any functional failures of the instruments must be investigated by the technologist and resolved as quickly as possible. Microbiology, the department of the lab that analyzes and identifies microscopic organisms from patients suspected of having an infection. Perhaps the most diverse types of samples are seen here, such as blood, urine, stool, swabs, body fluids, and tissues. In order to have enough bacteria to work with, samples are initially smeared or streaked on agar plates, also called petri dishes, which provide bacteria the necessary nutrients to rapidly grow. The setups bench is responsible for this initial step. There are multiple types of agar plates, some that select for the growth of specific organisms, some that inhibit the growth of certain organisms, and some that turn different colors based on what organism grows. Using multiple types of plates helps the technologist isolate and identify organisms. The streaked plates are then placed in an incubator and will be analyzed in 24 hours. Certain types of samples require a gram stain to be performed as one of the first steps. A gram stain provides a rapid and preliminary result as to whether any bacteria are seen in the sample prior to being grown for 24 hours. To perform a gram stain, a drop of sample is smeared on a glass slide. The slide is flooded with purple crystal violet stain, then rinsed. Iodine is then added to the slide. Then rinsed off. Then this slide is rinsed with the decolorizer. And again with water. Finally, a pink counterstain called safranin is added. and rinsed off. Once the slide is dry, it is ready to be looked at by a technologist. Bacteria that stain purple are called gram-positive, and bacteria that stain pink are called gram-negative. They stain differently due to their outer structure. Bacteria can appear in a spherical form, called cocci, or in a rod shape, called bacilli. Gram-positive bacteria can be seen as a cocci in chains, cocci in clusters, pairs, tetrads, rods, chains of rods, palisades, branching, or filamentous forms. Gram-negative bacteria can be seen as a diplococci, rods, branching, or filamentous forms. The technologist reports the morphologies of any organism spotted, as well as the presence of neutrophils and squamous epithelial cells. Any organisms found in cerebrospinal fluid are considered a critical result, and a phone call must be made to the provider. After 24 hours of incubation, the plates are ready to be initially looked at by the scientist, and any preliminary identifications are made now. The technologist notes the shape of the bacterial colonies, color, hemolysis, texture, and even smell. Believe it or not, smell can help identify organisms. Some organisms have a fruity odor, such as Pseudomonas aeruginosa. Some have the smell of butterscotch, such as Viridin strep. Some have a specific odor, such as E. coli. While others have a rather pungent odor. There are numerous biochemical tests that can also be done on the bacterial colonies. For example, a classical test to differentiate between Staphylococcus and Streptococcus species is the catalase test. A drop of hydrogen peroxide is added to the bacteria. If a sizzling reaction occurs, then Streptococcus can be ruled out. A wet mount is a simple but useful microscopic test that looks for motility in bacteria. Bacteria that have flagella will appear as clear particles, moving in a linear fashion, in circles, or spinning like a propeller. Some microbiology labs may get a rapid identification of bacterial colonies by utilizing a Maltitov, which stands for Matrix-Assisted Laser Desorption Ionization, Time of Flight. The Molotov is a complex modern instrument that zaps the bacteria with a laser into tiny particles, which travel up a column while sensors track the particle's time of flight. The software gives its best match as to what the likely identification is, based on known databases. If the specimen submitted is a set of blood culture bottles, a special instrument is typically used to detect bacteria within as little as a few hours. Blood tubes are loaded directly onto the analyzer. Once inside, the sensors can detect the presence of bacteria by measuring the CO2 level inside the bottles. If the CO2 level begins to rise, the machine begins to alarm, and a technologist quickly unloads the positive blood bottle. A gram stain is performed, and the blood is smeared on agar plates to grow the bacteria and eventually identify it. Antimicrobial susceptibility testing is an important part of microbiology and is often done via the disc diffusion method. In this method, a colony from a plate is subcultured onto a second plate that has a series of paper discs soaked in antibiotics. As the bacteria grow in the incubator, a zone of inhibition is seen around the discs. This zone is measured in millimeters, and the bacterial susceptibility rating is reported based on the zone size. Besides bacteria, microbiology staff are also trained to look for other microorganisms. such as parasites and fungi. Blood bank, the critical department of the lab that collects, tests, and prepares blood products for transfusion services. A decently sized inventory of blood products exists at most blood centers and includes red blood cell packs, plasma packs, cryoprecipitate packs, and platelet packs ready on demand. Each unit is kept under a strict temperature range and in correct storage conditions. The fundamental and most important test in blood bank is the type and screen, which consists of an array of tests that determine the patient's blood type and if any antibodies of concern are present in their serum. Many labs have an advanced instrument capable of performing this test, but we will be discussing the tube method, which every blood banker must know. The first step of a type and screen involves centrifuging the tube of blood. Aliquots of serum or plasma are made, as well as a suspension of red blood cells from the bottom of the tube. In the next few steps, the technologist will be performing a series of tests called the front type, and a series of tests called the back type. In the front type, a solution of anti-A antibodies is added to a small amount of the patient's red cell suspension. Next, anti-B antibodies are added to a second tube of cells. Then, a solution of anti-D antibodies is added to a third tube of cells. These are then centrifuged.
Shabloop, shabloop.
The technologist then gently agitates the cells on the bottom of the tube and tilts the tube on its side while checking for any signs of cell agglutination. If any agglutination is noted, the technologist assigns a severity score to the agglutination reaction. If the red blood cells have the A antigen on their surface, then the anti-A antibodies in the solution will bind these cells together, forming visible aggregates in the tube after centrifugation. The same principle applies for B antigens and D antigens on red cells. Once the front type is done, we now know the patient's blood type, denoted as A, B, AB, or O. This means that if the A antigen is present, then the blood type is A. If A and B are both present, the blood type is AB. If neither are present, the blood type is O. If the patient also has the RhD antigen, we add a plus after the blood type. In this case, the blood type is said to be O positive. The back type is performed to double check the front type. This is a reverse reaction that involves adding type A red cells to an aliquot of the patient's serum and adding type B cells to another. The tubes are spun down and likewise checked for agglutination. Thus, an individual who has the A antigen and lacks the B antigen has anti-B antibodies naturally occurring in their serum, and their serum will react with B cells, but not with A cells in the back type. In the next step, called the antibody screen, the patient's plasma is mixed with type O red blood cells with a list of known surface antigens. The idea behind this test is that if the patient's serum contains any other antibodies besides anti-A or B, they will bind to the reagent red cells and will cause them to agglutinate. This is made possible by adding anti-IgG antibodies, which bind together the tails of the patient's antibodies and makes reactions visible. It is very important that prior to adding anti-IgG antibodies, the cells in the test tube are washed. This step removes most antibodies from the mixture and only leaves the ones bound to the reagent cells. If no significant antibodies are present, then the screen will be negative. However, if agglutination is observed with any of the screening cells, this would mean the patients may have an unexpected red cell antibody in their serum. The technologist notifies the provider of this finding.
Yeah, we found it.
The type N screen now reflects this to an array of tests called an antibody ID, which attempts to pinpoint the exact antibodies responsible for causing the reaction. The technologist is now tasked with solving a large, complicated puzzle, which may quickly turn into a big mess. An antibody ID requires testing a panel of reagent red cells with a known antigenic phenotype against the patient's serum. Using the obtained reactions, the technologist rules out antibodies and is left with the answer. Things are further complicated if the patient has autoantibodies, as additional testing may be required. Freshly collected blood products are typed and stored in their appropriate locations and may also be distributed to other blood banks. When a transfusion is scheduled, the staff tests the patient's serum against the red cell products, making sure no agglutination is observed. Once testing is complete, blood products are issued out. Often, people do not understand why blood must be drawn for a type and screen prior to each transfusion. A natural question is, why can't we just grab blood off a shelf and give the patient's blood type to them? After all, it seems like such a waste of time running a type and screen frequently, right? Not quite. Suppose we have a patient, Bob. Bob's blood type is A positive and he needs an RBC transfusion. Bob gets his blood typed. His blood is cross-matched with an ABO compatible RBC unit, and the unit is issued to Bob for a transfusion.
I'm gonna monitor your vitals here, okay?
Suppose Bob receives a red cell unit that is positive for the Duffy B antigen, but Bob's blood does not have the Duffy B antigen. Bob's immune system now develops antibodies to the foreign antigen. Suppose Bob needs another transfusion a few weeks later. He is transfused another ABO compatible unit that happens to also be positive for Duffy B antigen. The moment the new RBCs enter Bob's circulation, his immune system immediately attacks the Duffy B positive red blood cells, causing a potentially catastrophic hemolytic reaction. This is why each transfusion needs a fresh type and screen, so that any new antibodies can be detected since the last transfusion. In cases of extreme blood loss, such as trauma, a mass transfuse protocol or MTP may be activated. The blood bank receives the activation warning on their pager and must be ready to provide uncross-matched blood products on demand. Because this is an emergency, the blood issued out is uncross-matched type O blood. The blood bank also performs testing for fetal bleeding in Rhd negative mothers who are carrying an Rhd positive baby. Normally, fetal blood is kept separate from the mother. If the baby's blood gets into the mother's circulation by mistake, the mother may consequently develop anti-D antibodies, and any future pregnancies will likely result in the baby having a deadly hemolytic disease. Thus, proper medical intervention relies on this test result. After adding the proper reagents, the technologist performs a microscopic observation of the reagent red cells and the mother's cells. Visible rosettes will be seen if fetal cells are present among the mother's red cells. It is worth briefly mentioning other clinical laboratory tests, some of which are done only in special reference laboratories. PCR, or polymerase chain reaction, is a molecular test method performed in many labs. This incredibly sensitive test can detect pathogens, such as a virus, in a sample. PCR works by amplifying nucleic acids, such as DNA, to detectable levels. Let's say our sample has a virus, which contains a DNA molecule. In the PCR method, the sample is heated until the strands separate. Short strands of DNA, called primers, are added to the sample. These strands are specific for a piece of code in the virus's genome and bind the DNA molecules. Next, a polymerase enzyme is added, followed by free nucleotides. The enzyme uses the free nucleotides to replicate the DNA molecule. By repeating this cycle several times, the amount of DNA in our sample greatly increases until it becomes detectable by the instrument. Some reference labs may perform ANA testing, which stands for anti-nuclear antibodies. Patient serum is added to a layer of cells. The scientist then adds fluorescent antibodies, which allow for the visualization of binding patterns inside of the cells. This part is often done in a dark room and using a fluorescent microscope. Possible patterns include speckled, homogeneous, peripheral, centromeres, and nucleolar. The purpose of noting any patterns is to help diagnose various autoimmune diseases, which often present as one or more of these patterns. For example, many patients with rheumatoid arthritis will have a speckled ANA pattern, or a homogeneous pattern. Other common test methods include gas chromatography, often used for drug testing, protein electrophoresis, used for separating serum proteins, atomic absorption spectroscopy, used for measurements of trace metals, rapid plasma reagent, used to screen for syphilis, flow cytometry, used to identify cell populations, often in leukemia patients, and many more methods which exist. The medical laboratory science profession is one that requires a careful eye to detail, ability to analyze data and solve complex problems, and a very good understanding of the human body. The goal of the scientist is to use their unique skills and knowledge to serve others, and to help save lives through diagnostics.